I just got a nice walk down Sunset Boulevard, so I'm all warmed up. I'm super excited to give you some updates on what we've been up to at Retro. I'm Joe Betts-Lacroix. I'm a serial founder, entrepreneur. I'm running a biotech called Retro, which is working on coming up with therapeutics to extend healthy human lifespan. And our mission is to go for 10 years broadly for people. Specifically to the most recent point, we're not trying to do that entirely just for billionaires. We want it to be broadly accessible, and that's part of our mission criteria for every program that we create. So just a little update on where we are now. We're running 4 therapeutic programs or pursuits. Been running just about 4 years now, and we're in the mid-70s people-wise. We have a lot of in vivo work. We believe in a lot of vertical integration. We've raised a bit of cash so far, and we have a couple of beautiful buildings over in Redwood City that are packed with labs and smart people. So probably in this audience, I don't need to belabor the point that aging sucks and destroys basically 100.00% of everybody on the planet. Here's this very depressing and captivating sequence of self-images by an amazing artist as he's starting to go down over a multi-year period from Alzheimer's and then eventually dying. We have to do something about this aging thing. At Retro, we're dividing this in big chunks across these 2 groups of either rejuvenating the cells that exist already in our bodies or essentially kind of giving up on them and just replacing them with young cells. More about that in a second. So we have, as a rejuvenation, especially our autophagy program, which will be going into the clinic in a few weeks, which is exciting and nerve-wracking, of course, and also a research program in tissue reprogramming that is not quite ready for the clinic direction yet. And then on the replacement side, which are 2/3 of our clinical programs, because I think right now it's a lot easier to make replacement work than rejuvenation work, because rejuvenation, you have to understand how a cell works, and cells are super complicated still to our tiny 2025 brains and computers. But now we know how to make young cells, and there are some cells that you can actually replace in the body, and let's do what we can right now. So the two of the programs are going after Alzheimer's disease as an example indication, essentially. I don't mean to put that lightly, since it's one of the worst, scariest, most horrible, soul-destroying manifestations of aging, but it is only one of them. And so the 2 programs we have that are a small molecule drug called RTR242 that restores the autophagy process that occurs in Alzheimer's, in a late-stage neurodegenerative neurons, in Alzheimer's and a few other horrible diseases. And then RTR888, which is working on replacing microglia in the brains of humans. And that is because age-related inflammation in the brain is a hallmark of a number of different age-related diseases of the brain. And getting the old microglia to stop being inflamed seems like a very hard problem. So at the moment, we can just replace them. So I'm just gonna skip through a few slides here, kind of describing some of our recent data in these couple of programs I'll touch on, because it's a very short talk. But I'll be around for a few minutes in case you want to ask me more about them. So for RTR242, the autophagy process is a natural phenomenon in every one of our cells that maintains this sort of cycle of recycling. Old and broken proteins, toxic protein waste that builds up in the cell is broken down and turned back into amino acids. This gets stuck in Alzheimer's, and gunk builds up in the cells. And this is just showing here on the left is the result from an in vivo, in other words, in a mouse model of Alzheimer's, study showing that one of the clinical markers people look at for human Alzheimer's is partially restored by a reasonably short amount of dosing of the mice, which is the ratio of amyloid beta 42 divided by 40, which is the sort of chain length. And that's going to be starting a phase 1 in Australia in a few weeks. And then the other therapeutic for Alzheimer's is a microglia replacement in the brain, which is, I know it sounds a little scary. It kind of is. It's a bit sci-fi that you can actually like change, about 10% of your brain cells are microglia. They're the resident immune cells of the brain. You can kill them all and regrow them, and they migrate and replicate inside your brain. Try not to visualize that too much, but it's actually a benefit because it means you can replace them. We have proofs of concept of replacing them in mouse brains, and we can generate new young ones in the lab, which we've done quite effectively. And then a third clinical program right now is also a replacement, and it's replacing the cells that make all of our blood. They reside in the bone marrow. They're called HSCs. And this is just a snapshot of just a few of the different blood types, but there are many, many blood cells that are all produced by these hematopoietic stem cells in the blood. They produce about 80% of the total cells, depending on how you define a cell in the human body, of the human cells, many of which are turned over in periods of hours, days, or weeks. So what we do there is make new hematopoietic stem cells starting from pluripotent stem cells that are derived from the patient themselves. And then we test them by putting them in the bone marrow of these specially immunocompromised and then humanized mice, and then make sure that they engraft over long periods of time, because that's one of the difficult parts, and then measure in the mice later to make sure that the human cells are now developing in these mice in the reasonable ratios that would indicate that they can be a life-sustaining set of blood for a new blood for a human. So one of the questions for this is, okay, you've made these young HSCs in the lab, you've put them back into an organism, we'll be putting them back into humans in the not-too-distant future. Why do you think they're young? So in the aging space, it's essentially a truism. Now, it's fairly well established that you can take old cells and fully reprogram them back to pluripotency using Yamanaka factors is the canonical way of doing this. And almost all of the aging characteristics of these cells are reset. And one way to measure this is epigenetic age, of course. And so you can see, on the left we have various donors of the cells that are used as the input to make these pluripotent stem cells that are then turned into the blood stem cells at a range of ages. And then when they're reprogrammed to iPSCs, they're essentially 0 age. That's the sort of inside the dotted box there. And then when so here's a zoom in of the dotted box. You can see when they're iPSCs, by the epigenetic clock, it's not super accurate around 0 age, so it shows them as being negative a few months. But one of the concerns we had is, okay, we run them through this fairly long, arduous, detailed differentiation process to convert them from being pluripotent stem cells into HSCs. Does that re-age them back to the age of the donor? But what we see is that only adds a few months, at least by these epigenetic clocks of age. And then what about once they regraft into the bone marrow? Does that re-age them? And that, at least in mice, not much. So essentially, it seems as though you can get a set of young blood flowing through your veins after replacing some HSCs. Don't hold your breath. It's probably going to be many months before we get to try this in humans. But hopefully we create a huge amount of additional healthy life. So I mentioned that it's pretty hard for humans to figure out how to rejuvenate cells, hence so much emphasis on replacement for us. Cells have 20,000 genes, and there's a lot of gene-gene interactions. Humans are kind of crappy at doing these sort of visualizations in their head of, let's see, what is the interaction between 20,000 times 20,000? How does this whole thing work? So we need some kind of computational tools to model the whole thing for us. One road we went down for that to try to understand like how to do anything new in biology, because most of the things that people have done in biology that are very transformative are kind of using old things. And for instance, like we have PCR, and people use it in every single lab pretty much every day. How did that come about? Somebody thought, hey, we could use enzymes that already exist in some extremophile bacteria and use them in the lab. Why do they have to go to those great lengths? Because it's really hard to just come up with your own proteins and your own enzymes. Pretty much right now we have to borrow them from existing slots. Or similarly, CRISPR is another enzyme. It'd be great if you could just say, hey, I need an enzyme to do this purpose. It's physiologically, chemically possible, design it for me. But we can't really do that. So CRISPR is another thing that's sweeping through and revolutionizing biology and probably medicine reasonably soon. And that's another bacterial enzyme that we borrowed. So what we asked was, could we start just designing our own proteins instead of having to borrow them from biology? And so we started off into this sort of, I said, like a joint research project with OpenAI, who's good at making big language models. Turns out proteins are essentially a kind of language. They're a sequential data type, right? So text, has letters that go in a certain order, and that gives it its meaning. And proteins are also arranged of a linear sequence of amino acids. So it's possible to kind of rebuild a large language model around that, which is what we did with them. We started with the scaled down version of GPT-4, and then retrained it with a lot of native protein data. And because reprogramming is a big part of a lot of what we do, we decided to test it out. It was built sort of as a generalized protein generator, but we decided to test it out on proteins that are near and dear to our heart, which are these Yamanaka effectors I mentioned, that can reprogram cells back to pluripotency and rejuvenate them. So we tried making some new versions of these that we wanted to be more potent for reprogramming, and some of them were extremely effective at that. It made reprogramming more efficient, because reprogramming isn't super efficient. It works, which is amazing, but it also works less and less the older the cell is to start with. And since our entire mission is arranged around helping older people, and we want to reprogram the cells of older people, coming up with variants that work better in age are important. So that is what we did, and it was shockingly effective to us. We're now embarking on this sort of next cycle. So we have these 3 programs I mentioned to you that are going toward the clinic, and now we plan to launch a bunch more based on money that we're raising right now to expand what we're doing. And we'll keep doing that on these 2 threads of replacing cells, because there's a few more that can be replaced, and also rejuvenating. So in general, our method of doing that is to make programs, and like I said, we have this sort of stringent program selection criteria. They have to ultimately be scalable to large populations, otherwise we don't start the program. And our method of getting those out into the population is to start off with narrow, more severe diseases, and then expand the indications to a larger number of diseases, and then ultimately they have to be conceivably used preventatively. And here's the three that are heading to the clinic. The small molecule program, classic oral drug, pharma has made that very efficient in general. So that's definitely getting into the clinic first, sometime between now and the end of the year, and then the others will follow later. So that is what we're up to lately. There's a little snapshot. Happy to take any questions. I don't know how many minutes I have gone under or over, so let me know. Just cut me off when the question thing is over.
Joe, great talk, great presentation. I'm so happy that Retrobio is here to tackle these multiple aspects of aging and broad strokes, pioneering visions. So the question I have, I was blown away by the very first slide on the Alzheimer's disease, A-beta 42 to 40 ratio. Is it done in Alzheimer model mice? And if so, is it in their brain? And if so, is it by immunofluorescence, or how was this determined?
So I believe this was peripheral, and so the slide...
In blood? Is it circulating A-beta?
Yeah.
Okay, then, yeah.
Which is hard to measure because it's noisier.
Exactly. No, that makes it even more
It's easier to measure in like cerebrospinal fluid, but it's also really hard to get CSF in mice. So fortunately, we were able to see the signal in blood.
It's probably ELISA then, right? Probably ELISA.
Yeah, I could possibly put my foot in my mouth if I try to describe exactly which proteomic measurement technique it may have been.
Anyways, it's amazing.
Like a sandwich ELISA-ish thing that they do at MSD.
Yeah, that's what makes it even more important. My second question is kind of more, not negative, but critical. For hematopoietic stem cell replacement, of course, there were ideas about it for a while. Mm-hmm. But one of the problems is that they will not engraft until you make a niche for them in the host bone marrow, which right now requires like total body irradiation. So are you guys thinking about making it better?
Yeah, for sure. So the initial, it's like Irina mentioned, it's been a lot of work over the last 20, 25 years for people trying to figure out how to get these things to engraft. Sometimes they can get them to engraft, but then they're only making one type of blood. But getting this sort of holy grail of long-term engraftment, also you can get them to short-term engraft and everything looks great, and then 3 months later, no more blood. So this was finally cracked near the end of last year by a stem cell research team that had been working on it for 20 years, I think, in Australia. And so we licensed it from them because we'd been wanting to do this for a long time and didn't know how to do it ourselves. And it hits all these criteria. Then there's this question of, well, how do we replace the existing blood system that a human has? Because it's sort of awkward to have no blood system in between a time when you get rid of a blood system and put in a new one. Also, the ways that people get rid of blood systems presently is harsh. So fortunately for us, the whole process of swapping out blood stem cells in a human is fairly well established because it's one of the primary tools that we have in the treatment of leukemias. And so that's been developed, it's called a bone marrow transplant. It's been developed over the last few decades to be quite reliable in a lot of ways. But one of the things that often happens, that often has to be done to these patients right now is because they have a leukemia, which means some of their blood cells are going crazy and creating too many cells, which is what causes the health problems for these people. They will give them drugs or radiation that wipe out all of their entire blood system, which is very dangerous, and then replace it often with the blood stem cells donated by another person. And then they have to live on immunosuppressive drugs typically after that. So early on, we're starting off in indications where people, for some reason, their blood system has failed because there are a bunch of different things that will kill off your blood system. as an adult. It's relatively rare, but it happens. So in those cases, we don't have to worry about this process. And then there are some leukemias where people are already, unfortunately, they already have like a very terrible thing happening to them, and having one of these procedures that remove their blood stem cells isn't the worst thing that they're facing. But for the long-term picture, because, as I mentioned, all of our programs have to ultimately be used on large populations of relatively healthy people to help keep them healthy longer, we don't want to give somebody whole body irradiation or give them a harsh compound like busulfan, which causes lots of other ill effects. And so there are a number of methods that can create some space in the bone marrow niche. And so the idea is to use this therapy cyclically. So you give them a compound that is used presently for, like one example would be, there's a compound that's used for mobilizing blood stem cells of a healthy donor, so that they leave the bone marrow, go into the circulation, and then you can draw blood from the circulation, separate out the blood stem cells, and then give them to the recipient to save their life. So that procedure itself is relatively well tolerated by the donor, but creates some space in their bone marrow. So the idea is to use a process like that, give them an infusion of these young HSCs, which will then occupy X%, say 15% of their bone marrow niche, and then do a cycle like that again. So after a number of cycles, we don't know exactly what the number of cycles needs to be. Of course, we'll try to reduce it to the smallest number possible. Then substantially eventually all of their blood stem cells will be the young ones.
Any other question? Sorry, we're running out of time, so we're running late. But is there a question in the back or anything? Okay. Well, thank you, Joe. You'll be sticking around for a few minutes, so go grab him if you need to answer, ask him anything. Thank you.
Thanks so much. Thanks, Brenda.